Well, hi there. Welcome to part 2 of our cell membranes series of video lectures. So in the previous installment, we mostly looked at the physical structure of cellular membranes, which are based primarily on phospholipid bilayers. And then to enhance the overall function of membranes, various proteins are embedded within the phosphobilayer matrix, I guess would be a reasonable term to talk about it. And collectively, the phospholipid bilayer and the proteins give all cellular membranes a property of being what we call selectively permeable, sometimes also just referred to as semi-permeable. But I think selectively permeable is a much better term to use because cell membranes can often be very precise in determining what substances can cross from one side of the membrane to another, whether that's taking in molecules and nutrients, raw materials from the external environment and bringing them into the cell so they can be used for energy and to build up other molecules in cellular metabolism. Other times, cells generate certain molecules that they then need to release to the outside environment, whether those are waste products or whether those are valuable molecules that are then used by nearby cells or have some other function as a part of the entire organism. So one of the primary functions of any cellular membrane then is to control the transport of substances across membranes. So that's what we're going to talk about in these next couple of installments. We're going to talk about the different methods by which materials move in and out of cells. And when we look at the mechanisms by which things move in and out, we can largely describe the mechanisms as falling into one of two categories. Either substances can move across the membranes through what we call an passive transport process, or they can come in through an active transport process. All right? So active transport is when a cell actually utilizes its own energy reserves to power and force the movement of molecules from one side of the membrane to another. Usually this is being done because they're actually trying to move molecules against a concentration gradient. Passive transport is when the cell doesn't need to use its own energy. There is already enough energy available amongst the molecules as they are moving that allows them to move from one side of the membrane to another. And so we're going to look at these. So this particular installment that you're watching right now is going to focus on passive transport processes. And then we'll come back and we'll do another recording for the active transport processes. So passive transport. Passive transport really always occurs because of an underlying fundamental kind of property of matter, particularly when you're talking about matter that is either in a liquid or in a gaseous state. Okay? And that process is diffusion. Okay? All passive transport processes ultimately are the result of this process of diffusion. Diffusion is simply a very natural tendency for molecules of a particular type to spread from areas where they are very closely concentrated, lots of the same molecules, all close to one another, moving around and bouncing against one another, and they naturally spread from areas of high concentration to areas where there aren't as many of that particular type of molecule. Okay. The reason why diffusion works is all substances have an inherent amount of heat energy that causes molecules to just randomly collide and bounce against one another in different directions, right? Because they're in motion. That's one thing that heat energy does, is that heat is really the energy that keeps molecules moving around, right? And just about anything that we encounter here on Earth has heat energy in it. Even if it's cold and frozen, like ice, the molecules in ice, although they are at the freezing point, there's still enough heat energy in ice that the molecules are very slightly vibrating against one another. Of course, they are also being held into place by mutual hydrogen bonds, which limits their motion. And because the amount of heat energy is low enough, the molecules can't overcome those hydrogen bonds, so they don't go very far. And of course, that gives them a solid structure. And things that are solid tend to not diffuse very easily because there are enough chemical bonds and forces holding all the molecules in their positions relative to one another. But when you're talking about liquids and gases, those molecules have more ability to move around one another. And there's a little more heat energy in those substances that causes them, as they move, sometimes two molecules or particles will run into each other and bounce off each other and collide. And then, of course, if there are other molecules nearby, they're going to collide too. And they're going to go back and forth and back and forth. And the more heat energy you add, the faster they move. And the faster they move, the more they bang and the more they can move other molecules out of the way and speed things up. So substances that have higher temperatures are able to diffuse with greater speed. So this, again, this is an inherent property of all matter, but it's most visible in liquids and gases because those molecules are far enough apart and are not as strongly bound to each other that they have that freedom of motion to continue to spread further and further apart. So diffusion, again, is just something that molecules in those states very naturally do. All right. Now, if you're talking about substances that are somehow contained, that is, we have another substance that creates a barrier, so like a glass beaker, all right, or a cardboard box that's been sealed with air inside of it. All right. But if you start out, and maybe let's deal with this in a liquid, because we are going to be talking about cells. Cells are liquid environments. Best example that we can probably use. You've probably done this. Have you ever worked with food coloring? And you've ever put a drop of food coloring in a glass of water and just let the drop hit the water and not stir it immediately? All of those dye particles are concentrated initially. And then that drop settles down near the bottom, but then you begin to see the spread of color. Okay. And what happens if you leave that drop over the course of several minutes to maybe an hour or 2 hours? What's going to happen? Well, eventually those pigment molecules are going to spread from where they were concentrated all throughout the vessel until finally you get an even distribution. And the color does not show any sign of concentration in one part of the glass or another. This is referred to as equilibrium. And so at a certain point, the molecules are colliding and bouncing around back and forth, until you finally get to the point where the same numbers of molecules are in any given area at a particular time. And so we have an equilibrium state. Now, equilibrium does not mean the molecules have stopped moving. It's just that the movement of one particle in one direction is usually countered by the movement of another particle of the same type in the exact opposite direction. The net effect, those movements cancel each other out. Everything seems to look the same. All right. So diffusion is the starting point to understand passive transport. All right. What's interesting when you apply this to cells is that cell membranes are able to create specific compartments. Right? They create containers in such a way, and because of the selective permeability of membranes, can often create regions where a concentration gradient always exists, because molecules may diffuse from one side of that membrane where they're more concentrated, diffuse across the membrane to where they're less concentrated, but then as they get inside that membrane compartment, those molecules are chemically consumed or altered so that they're no longer that type of molecule. And that's the thing about diffusion. Diffusion works separately on every different type of molecule. All right. You actually probably observed that. If you have performed the diffusion and osmosis lab by the time you've seen this video, you remember in that experiment that when you put a glucose solution inside the dialysis tubing bag, right, there was diffusion of glucose from where it was highly concentrated inside the bag across the membrane of the bag to the beaker fluid on the outside. But then there was also diffusion of water from outside the beaker into the dialysis bag. So glucose and water are two different molecules, and they both diffused down their concentration gradients. And now if we had some kind of a chemical process that as glucose was diffusing out of the bag, and we somehow removed the glucose or turned the glucose into another kind of molecule, then we could continue to pull glucose out of the bag, and we might never get to an equilibrium point between them. And that's generally what happens in cells. So like when cells take in oxygen that they use in the process known as cellular respiration, there is always higher concentrations of oxygen outside of the cells. And then when oxygen diffuses across the membranes into the cell, that oxygen is consumed to provide energy within the cell, and the oxygen is used up. And so there's always less oxygen in the cell than there was outside the cell, because metabolism changes the natures of these substances. And it can work the reverse. When carbon dioxide is created in cells, CO2 is a higher concentration inside the cell than outside, and so the CO2 tends to diffuse out. And because metabolism is always generating new CO2 inside of that cellular compartment, we will always have continuous diffusion, and we won't necessarily get to an equilibrium state. Okay. Now, since we're talking about diffusion, and I just recently brought up the osmosis experiment from your laboratory activities, let's talk about osmosis, okay? Because osmosis is a very specific case of diffusion, right? And it's really, really important in the functioning of cells. Right? Osmosis specifically refers to the way water molecules diffuse between solutions that have been separated by some kind of a selectively permeable membrane. But that membrane must be permeable to water, but is not necessarily permeable to other dissolved molecules in either one of those solutions. All right. So just to remind you, a solution is a mixture. Okay? It is a fluid that contains both a primary liquid molecule that we refer to as the solvent, all right? And in biological systems, water is the solvent. But then any other molecules that are mixed into that liquid are referred to as solute. So when we're looking at cells, cells have different types of solutes inside themselves, in their cytoplasms, than exist in the fluids outside of those cells. And so water can often diffuse between different areas depending on the relative solute concentration. Right? Cells have these different concentrations of solutes compared to that. And it's really the job of cellular membranes to maintain those differences in solute concentrations. Because again, the cell is manufacturing and consuming different types of molecules as part of its metabolism. There is a very unique chemical environment inside the cell that is always very different from what's going on outside of the cell. All right. But when it comes to osmosis, it doesn't matter necessarily what type of solutes there are. All that really matters is the relative concentrations of solutes between the inside and the outside environment. All right. But let's kind of start with something simple here, and let's understand how osmosis works. All right. So osmosis, again, is defined as diffusion of water across these selectively permeable membranes to areas that have a low solute concentration into an area with a higher solute concentration, and osmosis will continue until an equilibrium state can be achieved. So let's show you an example right here in this illustration. So here we have this kind of interesting U-shaped tube, but we have separated the two halves of the tube by a selectively permeable membrane with kind of like that dialysis tubing where you have tiny little pores. But in this case, the pores are just large enough to allow water molecules to pass through the membrane. But the larger concentration of a sugar molecule looks like we're dealing with sucrose, which is a disaccharide molecule. We have different concentrations of sugar. On the left side of the tube is a 5% concentration of sugar versus the right side of the tube, which is a 10% concentration. So we have double the concentration on the right side of the tube. Okay. So, of course, what does this really mean? Well, this also means that if there's a difference in the amount of sugar, that also affects the relative amount of water molecules. So if we're talking about a 5% sugar solution, so if 5% of the molecules in the left side of the tube are sucrose molecules, well, that means that 95% are water molecules. But then if you look at the fluid on the right side of the tube and on this side of the membrane, this is a 10% sugar solution. And so if 10% of the molecules are sucrose, that means that the other 90% are water on this side. So there is a concentration gradient between the two sides. So that means that as the water molecules, they will diffuse from where they are more concentrated to where they are less concentrated. And so as molecules are colliding and bouncing around back and forth, those random collisions of water molecules are going to be able to send water molecules through the pores from the left side over to the right side. And we're also going to get the movement of water molecules a little bit from the right side back to the left side. Okay. However, we're generally going to get more movement from left to right than we go from right to left. I mean, any molecule that goes from the right side to the left side is counteracted by ones going from the left to the right. But since we have more water molecules to work with, on average, more are going to make that transition. And so we're going to end up with an actual movement of water molecules from one side to the other. And as we add more water molecules to the right side of the tube, we do actually begin to affect the ratio because we are physically adding more water molecules. Suddenly the proportion of water to sugar begins to increase on this side. And of course, with the loss of water molecules from the left side, that proportion is also going to change, and we're going to have a proportionate decrease in the amount of water on the left side. And so we can see is that if we allow this to reach an equilibrium state, there has actually been a drop in the level of the fluid in the left side of the tube. And you can see that they started out equal volumes on both sides, but look what happened. We had a drop in fluid pressure on the left side, and we had an increase in fluid on the right side. And as a result of it, we changed it until we diluted the 10% sucrose solution by adding more water molecules. That became more and more dilute, and we went from 10% to 7.5% on this side. But then the loss of water molecules concentrated the solution from 5% sugar also to 7.5% sugar. And now that we have equal amounts of water on either side, and we have equal concentrations of sugar. So this is our equilibrium state. So this is really important because cells always have these selectively permeable membranes that can prevent the movement of some solutes from one side to the other. Now, why does this happen in cells? Well, it's because almost all cells create very special protein channels in their membranes called aquaporins. And aquaporins are basically almost like tiny little protein tubes that have a channel that is just the right size and shape for water molecules to pass through it, but other materials don't make it through. The thing with aquaporin channels is that they're essentially open in both directions. So water will always move across aquaporin channels in accordance to their concentration gradient. So wherever there's more water, they're going to travel through those channels to where there is less water. But what's really driving the difference in water amounts on either side of a membrane? It's the relative concentration of solutes, okay? Because cells may not necessarily have channels for particular solutes, or if they do have channels for a solute, often those channels can only allow the molecules to pass in one direction. Again, it has to do with the configuration and the specific shaping of that channel that may only allow that substance to go in one direction across a membrane. So if it diffused into a cell, it may not be able to diffuse out through the same channel due to specific reasons. All right. But what is it that really drives osmosis? Well, osmosis is really driven because the percentage of water in any particular cellular compartment, or even in the cell versus outside of the cell, is determined by the overall solute concentration, because those solutes are then able to generate what we call osmotic pressure. Okay? And we tend to think of pressure as this force that pushes in on things. And so why do we use that? Because we can really more describe osmotic pressure as a force that actually helps to pull water molecules across membranes into areas that have more solutes present to it. So we can think of solutes as kind of attracting water molecules into them. And the reason we use the term osmotic pressure is because to measure it, what we're imagining is that, well, there's this natural force that is drawing water from an area with low solute concentration across to an area with a higher solute concentration. All right. But how do we measure that force? Well, it's like there's a current pulling and water flowing through those channels. What if we wanted to prevent water from doing that without changing solute concentrations? Well, we could try to push the water back against it, but how much force would it take to push water molecules against their natural flow? Well, that requires a form of physical pressure to push against it. So we are able to measure the force at which water is flowing into it by imagining, well, how much force would be required to counter the natural flow? And so that's why we call it osmotic pressure. So osmotic pressure is this force that is generated by the presence of solutes. The more solutes you have, the higher the pressure there is. And that's what gets water to move across aquaporin channels and into cells. If you want a really simple way to keep that concept in mind, I've got a 3-word phrase for you that, as you continue to study and learn about osmosis and the implications of osmosis, use these 3 words to help you keep track of where is water going to go in the event of a difference in solute concentrations. It's this: water follows salt. Okay. Now, I don't literally mean salt all the time, although sodium chloride is a frequently set of dissolved ions that are present in biological fluids, and especially in the fluids that exist outside of cellular membranes. There tends to be the extracellular fluids around living organisms tend to be a little saltier in the traditional sense than what's going on inside. Okay? And that's because most cell membranes are very selectively permeable about sodium and chlorine ions and manage their locations. But let's open this up a little bit. When we use the word salt, using little quotes around it, really we're using that as a shorthand for any solute. So you could say water follows solutes. And wherever there are more solutes, water will diffuse via osmosis across membranes to those locations. So let's kind of put that to work here. Let's actually work through a simple example. All right. So here we have kind of a very simple model of a quote unquote cell made out of a selectively permeable material. And inside of our cell model, we have put in a solution that is a mixture of 0.03 molar sucrose plus 0.02 molar glucose. And then we suspend this little sac containing this mixed solution into a beaker that also contains the mixed solution, where there is a 0.01 molar concentration of sucrose, 0.01 molar concentration of glucose, and a 0.01 molar concentration of fructose. All right. So, so what do we do if this membrane is selectively permeable, and it will allow glucose and fructose molecules to pass across their surfaces, but sucrose is prevented from diffusing? All right. Well, let's just handle the diffusion of the sugars first, and then we're going to come back and we're going to see, well, and what does this do to the water portion of these solutions? All right. But first, let's look at the diffusion of sucrose. Now, where do we have a concentration gradient for sucrose? Is the concentration gradient going to have sucrose go from inside our cell to the outside environment, or are we going to have sucrose go from the outside to the inside? Trick question. Not permeable to sucrose. Doesn't matter that there is a concentration difference for sucrose between the outside and the inside. Sucrose cannot cross the barrier, so we will not get diffusion of sucrose across it. So we can ignore that part. Glucose, on the other hand, glucose can pass across the membrane, but what direction is glucose going to go? Well, there's more glucose inside the cell than outside, so we are going to get diffusion of glucose from inside the cell to the outside, and it will continue to diffuse until we reach an equilibrium state. But where is that equilibrium state? Well, it would be the halfway concentration between these two. So glucose would diffuse out until we change our concentration by another 5/1000ths molar, because the difference between 0.02 and 0.01 is 0.015, right? So that's our concentration gradient for that. And so once we hit equilibrium, we should have equal amounts of glucose on either side. Now, what about fructose? Well, there's only fructose in the outside environment. There's none in the cell. So that concentration gradient is going to have fructose diffuse into the cell. But then again, how long will that happen? Well, until we hit equilibrium. And so we begin to lose fructose and add it to the side. We will also get a little bit of a change here, but we're going to change this. When we get enough diffusion and we hit equilibrium for fructose, we will be adding those molecules into here. Okay, so these molecules will diffuse until they hit equilibrium. Now, what does this do to the overall water balance of our cell? How is this going to affect osmosis? Well, it doesn't matter what all of the individual concentrations are. As far as water is concerned, we have to add up the total solute concentration to see what's happening inside the cell, and then compare that to the total solute concentration outside of the cell. So if we add these together, well, 0.015 plus 0.005, so that's back to 0.2 plus 0.3. Our total concentration in the cell, even at equilibrium for that solution, for those sugars, is 0.05 molar. Now, what about these guys? Well, again, if we add 0.005 plus 0.015, that's 0.02 plus another 0.01. Total concentration in here is still 0.03. All right. So where is water going to flow? Is we're going to lose water from the cell and add it to the beaker? Or are we going to add water from the beaker into the cell? Well, water follows the solutes. Where do we have more solute? We have it inside the cell. Water will begin to enter our cell and begin to dilute both of these down. And when we hit an equilibrium state for water, then we're going to end up diluting what's inside the cell and concentrating what's outside the cell until the total concentration of sugars is at the equilibrium point halfway between these two, which is going to be a 0.04 molar solution. But we're going to add water to that cell. Okay, so that's an application of understanding osmosis. But this then has real life implications for cells. Cells can sometimes utilize osmotic gradients to help themselves out, but most of the time cells actually want to avoid experiencing any osmotic gradients or osmotic pressure. It really depends on the type of cell and how it's built. So if we were to take something like a animal type cell, like a red blood cell, okay, animal cells do not have external cell walls or anything to reinforce their physical structure. They just have that very flexible how would we say, fluid membrane. Remember, the phospholipids are huddling together, but they are not strictly bound to one another. Mm-hmm. And so they are technically the fluid mosaic model of cellular membranes. That's all that's defining the boundaries of this cell. So it's actually not very strong. So depending on the type of environment these cells are in, this could have consequences for animal cells in particular. If they are in the kind of solution that's referred to as a hypotonic solution, hypo means below or lower than. Tonic refers to the tension, okay, which is, tension is kind of it's sort of the opposite of pressure. If pressure is a force that is pushing things together or pushing against something is what we think of as pressure, tension is like a force where things are being pulled apart from one another. All right. So what's interesting, a hypotonic solution doesn't have a lot of solutes in it. So there's no tension, because without solutes to pull water in that direction, there's no tension in the fluid outside of the cell. If anything, the tension is created inside the cell because the cytoplasm has all of these solutes in it, and more solutes than what we have outside. So this causes osmosis to pull water into the cell, filling it to the point where the cell can eventually stretch and lyse, meaning that it breaks apart. So this is a huge problem for animal cells in that they can't be exposed to solutions that are too low in solutes. Otherwise, the cells will all swell up and you could possibly rupture those plasma membranes, which causes the cytoplasm to leak out. The cell dies. So this is a problem that can happen if people become overhydrated. And that can happen. People can drink too much water, fresh water, which as it's absorbed into the bloodstream and distributed around the body, actually causes cells to take on too much water, causing cellular damage. Right? And so people have died from over-ingestion of too much water too quickly. Right? What cells want to experience is an isotonic solution or an environment. Iso means to be the same, so that the level of tension inside the cell and outside the cell is equal. So any diffusion of water into the cell is matched by diffusion of water out of the cell. And so this is an equal balanced state. This is something that in animal bodies, they have systems to try to actively manage their extracellular fluids to keep them as isotonic as possible so the cells are not stressed out. On the other hand, if that comes out of balance, the opposite direction is to be in a hypertonic solution. So hyper means beyond or higher than. So there's a higher tension in the solution, meaning there's more solutes in the solution outside the cell than inside. If there are more solutes outside the cell, then that causes water to be pulled out via osmosis, and the cell loses water content, which causes it to shrivel or shrink. And specifically when describing red blood cells, when they do this, we can also use the term crenated. Okay, it's kind of like what happens to a grape when it becomes dehydrated and shrivels down into a raisin. So this is what can happen if you were to ingest a lot of fluids that have heavy solutes in them, really, really sugary solutions, or salt water for that matter. I mean, this is why people who accidentally get experience shipwrecks and lost out at sea somewhere can't drink the seawater around them, because the high levels of salt in seawater, if you drink them in, tends to pull water out of the cells. And why is that bad? Well, by concentrating all of the other materials inside the cell, the metabolism gets out of balance and cells can even die that way. So animal cells in particular are really sensitive to the changes in the surrounding fluids around their cells. Plant cells are a little bit luckier in that they have a little bit more structural reinforcement to help them out a little bit. They have an external cell wall, which can help protect some of the membrane. So if you put a plant cell in a hypotonic solution, that will indeed cause water to enter into the cell, and will cause the cell to begin to swell up until so much water builds in that it creates an internal fluid pressure that makes the cell turgid, or really, really kind of stiff. This is actually normal for a lot of plant cells, and this is what helps a lot of plants actually maintain their overall structural form, especially when you're talking green or what we call herbaceous plants that don't have natural wood. How do plants like that able to have stems that stand upright against the pull of gravity? Well, they're filled with internal fluid pressure because the cells are able to add enough water inside their cells to cause them all to swell up and make them stiff. All right? If cells begin to lose some of that water because they're not absorbing enough through their root systems, then they might begin to wilt, right? Because they're losing that fluid pressure as the fluids outside of the cells in the plant body become more isotonic. And so things become a little more flexible. Now the cells are still alive. There's still a good balance of water moving in and out of the cells, but the overall plant structure may not be as strong as it can be. On the other hand, if you put plant cells in a solution that's hypertonic, that's going to pull water out of those cells. And if you pull too much water out of the cells, the cell membranes can pull away from the cell wall, except in certain locations where the membrane is welded to the cell wall in strategic locations. But this causes the cytoplasm to shrink down, and we say that the plant cell has become plasmolized. And this is not necessarily going to lead to a functional cell. And if this persists too long, again, waste products and other aspects of metabolic function become impaired, and the cell might die. This is why if you pour salt water into soil around plants, they wither, because you actually pull the water out of the root cells to the point where they die, and without functional roots, the plants don't get extra water and nutrients, and they can't make it. Whew, that's a lot about osmosis. The thing about osmosis is that osmosis is just this special instance of diffusion. Now, diffusion is what happens as molecules move primarily across the phospholipid bilayer. And as we were just talking about, water diffuses in and out of cells because there are special protein channels that allow for the transport. Okay. And that's what makes it easier for water molecules to cross phospholipid bilayers. In case I've never done this before, PLBL is my shorthand for phospholipid bilayer, the normal material that cell membranes are made out of. Now, it turns out, depending on the relative permeability of the phospholipid bilayer, some water molecules can leak through those layers. It's just that aquaporin channels improve the efficiency for the movement of water, because water is polar and has trouble getting past the hydrophilic heads of phospholipid molecules. All right. Now, since we are talking about the diffusion of water and using a protein to aid in the diffusion, then we can describe this form of diffusion, and that osmosis is an example of not just diffusion, but technically it is facilitated diffusion, because the molecule is getting a little bit of help. Okay, help in the form of a protein that makes it easier for the substances to move across. As you remember, we talked about phospholipid membrane structure, and small polar and ionic particles don't get through very easily. But those are particles that often are needed by cells. And so protein channels work really well. And there can be all kinds of protein channels that provide a pathway, usually for a very specific type of molecule. Right? Now, some of these channels are like aquaporins. They are not what we call gated, meaning that they do not open or close. They are always open all the time, and therefore the molecules can move across them. Other channels, though, are gated, meaning that they do open or close depending on whether they have something to transport or not. Right? So one example of a gated protein channel are referred to as carrier proteins. These are proteins that actually change their shape only when the substance that they are meant to transport makes contact and kind of fits in against a specific contour of the carrier protein's surface or structure. And then that actual binding causes the protein to change its shape, allowing further passage of that molecule from one side of the membrane to another. So here's sort of a comparison of the two types. So channel proteins are usually just open tubes. All right. So the overall protein structure has a passageway that allows the molecules to pass from the outside into the inside. And this is going to, and the direction of movement is almost always down the concentration gradient. So there might be more of the solute that's passing through this channel outside the cell than there is inside. And so random collisions are generally going to send more molecules passing one way or another, or from the outside into the inside. Fewer particles are then able to make the reverse transit. Down here at the bottom are the carrier proteins. So a carrier protein has a shape which kind of makes it maybe open to one side of the membrane. A solute particle that this protein is meant to transport lands in a receptor site. So there's a specific contour to the shape of the protein that matches this particular molecule. Other molecules will not fit this shape, and they won't activate the carrier. It's only the shape of this molecule that allows it to bind. And once it attaches, the protein changes its shape to close the outer opening and instead open up to the inside and release the solute to the inside. This is a way a lot of important substances like carbohydrate molecules can be transported in this fashion. Glucose is transported this way. Glucose moves in by binding to a carrier protein, which then facilitates its movement across. Now, both of these models are still versions of facilitated diffusion because we're using a protein, but in both cases, we're not using any extra cellular energy to make these proteins work, because either they're a normal channel protein that just allows for the natural diffusion from high concentration to low concentration, or the molecule itself causes the protein to alter its shape to move it from the outside to the inside. But the net movement is always going to be from high concentration to low concentration. We differ facilitated diffusion from ordinary diffusion because ordinary diffusion is usually happening just through the phospholipid part of the membrane without the help of a carrier protein. So that's passive transport. So in the next video installment, we're going to take a look at, well, what about the movement of substances against concentration gradients? How can you do that? Well, the cell needs to expend some energy itself. So we're going to talk about a little bit about cellular energy, where it comes from, and we're going to talk about the use of that energy then in moving materials. So hope you have gotten a lot of information out of that this installment, and we'll see you in the next video. Thanks a lot.